Thanks so much, Nils. Yeah, and thank you all as well. This has been such a fun meeting. I usually go to a lot of biologically focused meetings. So it's been so cool to see people asking a lot of the same questions as us and coming up with a lot of unique methodological solutions, and especially to see some CODA data in the last talk. That was so cool. Yeah, so today I'm going to be talking a lot about PDAC and Panin, so I thought I'd give some biological motivation as well. PDAC, along with glioblastoma, is one of the deadliest malignancies in humans. PDAC specifically is deadly because it is difficult to diagnose until it's metastatic. So by the time patients come into the clinic, they often have distant disease and few clinical options. Because of this, a lot of focus lately has gone into study of the initiation of this disease. So PDAC develops out of two main known cysts that develop in the ductal system of the pancreas. One of those is called IPMN, which is physically big in size. It's distinguished by its size. Because it's big, it can be seen in diagnostic imaging like CT, but sadly, it takes into account the fewest PDACs. Most develop out of a second lesion called Panin, which is defined by its small size, making it at the moment impossible to detect in the clinic. So non-invasively, you can't know if you have any of these Panin lesions. You can't see it in CT and endoscopy, anything like that. So because of this, all we knew about Panin came out of histology. And just some statistics, again, PDAC is quite deadly. It develops out of Panin. As I'll show today, we have found with quantitative histological mapping that Panin is unexpectedly quite common in human tissues. All of us develop Panins as we age, which is a bit shocking because most of us don't develop PDAC. About 1 in 10,000 people gets PDAC, but as we'll see, all of us will develop tens, if not thousands, of Panins as we age. So why we knew so little about Panin is because due to its small size and its lack of symptoms in humans, it's often seen incidentally. So it's seen in histological tissue. Often patients come to the clinic and have a Whipple because of something else, and Panin is seen histologically in the same tissue. Because of this, because all we knew about Panin came out of 2D histological slides, the knowledge we had was limited by the medium, so it was two-dimensional. We didn't know anything about the connectivity, the incidence, and any field effect that Panins might be having on the adjacent tissues. So I'm going to do a quick pivot into a method. Because we wanted to study Panin lesions, we thought about developing a technology that was amenable to the disease. And I think that's kind of a way that we go about method development on the CODA team is instead of developing methods and then choosing a biology to ask questions in with those methods, we began with the question of studying Panin, and then we came up with methodologies that helps us study this disease. So because Panin is so easy to see in H&E, we began with H&E. And actually, most of the data that we analyze with CODA is H&E. We'll cut thousands of slides instead of tens of slides, build highly quantitative, almost old-fashioned anatomical maps of tissue, and then use a lot of advanced multi-omic techniques to ask pinpointed questions at specific locations in those maps. So to give a quick defense of H&E, I love H&E. I think we can get a ton of data out of one of the most classical stains in the clinic. If anybody doesn't know, H&E stains based on the pH of tissue. So hematoxylin is going to stain basophilic components like DNA. DNA is an acid, so the nuclei will stain in a deep blue. Eosin is going to stain acidophilic components like collagen and smooth muscle in a light pink. And then the cytoplasm of cells contains a lot of molecules with many pHs. So depending on the cell type, you'll get these shades of magenta and violet. So with just H&E, we can see cells and components of cells. And if we zoom out to kind of a mesoscale on the same slide, we can see tissue level anatomy. So now I'm showing a duct, and an anatomic pathologist in the clinic could tell you that the bottom of this duct at 6 o'clock is non-diseased epithelium, while the top half is neoplastic. It's those Panin lesions that we study. So immediately you can see how identifiable this disease is in histology. So let's come up with computational techniques that let us map it and quantify it. So a big focus in the lab is development of tools that make it easy to quantify histological slides. So this is an example of a segmented image and segmented at the tissue level, at the pixel level. So not nucleus segmentation, but pixel by pixel label of cell types. And what this allows us to do is identify many of the low-lying functional cell types that you could see when you look at a slide. So in this case, we have endothelium that lines blood vessels, we have smooth muscle, collagen, epithelial cells. In light blue is the islets that make insulin and glucagon. So without laying a single antibody on this tissue, we can identify, what is that, like 9 cell types immediately, and we can quantify those spatially. And because it's H&E, we can now go back to the tons and tons and tons of clinical tissue, scan that, and begin to quantify things at scale. So hopefully submitting sometime this week, a PhD student in my lab named Valentina has been developing tools that would allow anyone, even a bench scientist, to quickly and efficiently build highly customizable segmentation models. So with this, she's made a supplemental annotation guide that teaches people how best to annotate to quickly build a model. She's designed a GUI in Python that lets you customize the model, tell it how many labels you want to have, and teaches you how to customize these models to any tissue type. And to quickly show 2 applications of this, one application that we immediately thought of, when would you want to quickly quantify something in a pointed way? You have a specific question with a specific set of tissue. And one example of this is a mouse study. So say if someone's developing a syngenic mouse model and you have metastases popping up in many sites, if it's syngenic, you can't use flow to quantify metastases because they'll also be mouse DNA. So oftentimes, when I was a student, I was in a wet lab when I was a PhD student, and I would see students sit on ImageJ and count metastases and spend a month just quantifying something that's so distinct when you look at the image. So one of the applications of this GUI is allowing people to quickly build these custom models that could let them quantify things like metastases in multiple sites in a mouse. A second application is now moving into clinical histology. So just to give a quick motivation, CAPS is an initiative that was developed initially at Johns Hopkins, but now it's nationwide, that longitudinally follows specific populations of individuals at a heightened disposition of developing PDAC. So maybe they have some known mutation that is going to cause them to develop PDAC at 5 times the likelihood of the typical individual. So what we did is we took advantage of the unique data set that exists in CAPS, and this is being led by Lucy Duquette, a PhD student in my lab. And with these individuals, we get longitudinal endoscopy imaging that gives you kind of a meta-scale view of the acini and the fat and the collagen. But again, you can't see pannins. And with this, we get companion histology. So she's collected, I think, almost 5,000 now clinical histological images and built this highly customized segmentation model that, if you look on the left, this is what you and my pancreas most likely looks like. It's mostly filled with acini, which is the functional tissue that's making digestive enzymes. But one of these CAPS patients has a lot less acini. They have a lot of fatty deposition that you see in yellow, a lot of ECM. And this, Lucy has been showing quantitatively, is what we've been calling a field effect due to the development of thousands of pannins. And again, pannin can't be seen clinically, but what Lucy's showing is that if we can give evidence that these pannins exist, we can then build AI models to link that composition back to something that can be quantified in endoscopy images, and now build a diagnostic test that could let us follow these CAPS patients and tell them when we think they've passed that pivot point of having too many pannins. Okay, but that was all 2D, and this is not a 2D meeting. So let's move on to some new methods. So again, the segmentation, I think it's a highly usable component of CODA, but it's not the only thing that we do. It was just one step along the way to quantifying the incidence and the connectivity of these pannins. So the main concept of CODA was, again, we can see these lesions quite well in H&E. How can we come up with some combination of new technologies that let us image them and ask specific multi-omic guided questions? So what we do is we go back to the clinic, and we've been taking advantage of blocks collected all the way back into the 1990s. And instead of a typical study, say in the clinic, that you would cut just a couple sections, with CODA, we've been cutting up to 2,500 slides on a block. Because the medium we use is mainly H&E, it's much less expensive than some of the technologies that use cyclic codecs and things like that, that each slide can cost thousands. H&E is quite scalable. So we begin with these data sets, and then we've been developing computational techniques that enable us to align this histological data. And again, unlike many techniques that use tens of images, we need to make methods that can align thousands of H&E images and not get that skewing and pulling that some techniques can have when you accumulate many images in a block. So this data set, I think, is about 2,000 slides. So it's in physical size, it's about the size of Monopoly dice, is the analogy we like to use. We combine that with the segmentation that I just showed, which is done on a 2D basis. And with that, we can get these highly quantifiable tissue maps. And I skipped a slide about cell detection, but because we can also employ a lot of the existing methods like CellPose to do nucleus segmentation in H&E, we can combine the tissue segmentation that we get with CODA with any type of nucleus detection technology, and then label those cell types in H&E. So this is just spinning down many functional types. At the moment, it's the ducts and the blood vessels. Now you see the islets in light blue, the fat in yellow, the acini in violet, and then I think it's going to zoom in and show you single cell level. But just to justify quickly, why would we want to do this? I think this will be the second talk today that someone's mentioned why a TMA is not enough. And just using the basic compositional data that we get with CODA, we ask the question of, if I wasn't modeling this huge block of tissue that's the size of dice, if I was just taking a single TMA, a single whole slide image, how would my assessment of the tissue composition change? So in this specific question, we simulated a single TMA being taken at many angles in the same block of tissue, and we estimated what would be the malignant cell composition in each of those TMAs, and how many of these TMAs would I have to take, almost how many independent biopsies would we have to take to get a holistic sense of what's actually going on in the tissue. And what we found is it was shockingly many. I mean, to get to the actual composition, you needed almost thousands of individual slices. Second, in an analogous calculation, we wanted to know, okay, if I wanted to get an assessment of, say, pannin density, which is the main question in the talk today, how many sections would I have to take? And as you might expect, I think it's kind of intuitive, but it's always good to quantify these things. What we found is that in samples containing many pannins, you could estimate the incidence with just a few slides. But in samples with few pannins, so say we have a block that only contains 2 lesions, you needed many, many slides. And you can imagine why, because if you cut the slide without the pannin, you'll say you have none. And if you cut the slide with the pannin, the estimate will be too high. Showing that when we think about how much tissue do I need to sample, it's not a black and white question of 5 slides. It depends fully on the question you want to ask and how common is the phenomenon that you want to catch in the tissue. Okay, but going back to pannin. So the main question was, can we use this CODA technology, cutting many H&E slides, aligning them and segmenting tissues to map pannins? And the video that was just playing was showing you that we can follow now a pannin going down a block of tissue. And this video is an initial sense of what do they actually look like, because until this technology, again, we only knew what pannins looked like in 2D. So we knew they developed in a ductal system. So people thought they looked like discs, but what we showed is they could be quite complex. And I think of them almost like an octopus with all these tentacles following out into the ducts. This is a single pannin, but scaling up now to a whole block size. And this is actually the same block of tissue that we just saw in the last talk, which was kind of cool to see. The shocking fact 2, so if fact 1 was that pannin can be quite complex and have this unexpected connectivity, fact 2 was that in a single block of tissue, we found tens of pannins. We thought we would find 1 to 5. This block contained 57 lesions, defined by anatomical connectivity. And this was not an outlying sample. When we scaled up and we applied the same analysis to 50 blocks, we found not a single sample with less than 5 lesions. So this is something that just was not known because it's such a difficult tissue to model and a difficult disease to quantify because Pannin can't be specifically stained with a single antibody. It needed some kind of quantitative H&E method like this. And when we upsampled this, because again, we model big chunks of tissue with CODA, but it's still just a piece of something. So when we upsampled to the size of the whole human pancreas, we got a mean estimate that the population modeled in the study would have a mean of about 1,000 Pannins each, making this, based on what we know today, the most common lesion in the body. Do I think it's actually the most common? Most likely no. I think maybe it's the one that's been studied the most systematically like this, but maybe the colon would come close as well. Okay, so what I'd like to do now is step down this data set and give some examples of the many questions we've been able to ask with this initial database, is what I like to think of it, this database of anatomical maps that we built using only H&E. We've been able to ask questions that affect clinical classification and diagnosis, that affect how we think about Pannin initiation, and how we think about eventual development of PDAC by combining with things like Visium, things like IHC, and with spatial genomics. So beginning with a clinical question, at the beginning of the talk, I mentioned that 2 lesions exist, one called IPMN and one called Pannin, distinguished only by size in the clinic. So if the lesion is big, it's called IPMN, and if it's small, it's called Pannin, which sounds kind of shaky and non-scientific, but actually they've been shown to have 2 completely distinct outcomes. So it's meaningful somehow, even though it seems like a bit of an ambiguous designation. Well, what we found with the CODA data is not only do we all have lots of Pannins, but they can get quite big. And if we take one of these lesions and we cut it at 2 angles, so this pink line is an actual H&E slide, and then I can synthetically section this in the yellow line at any angle that I want to. And if I follow the exact alignment of the duct in this case, I can give you an image that in the clinic would have to be classified as an IPMN because it just exceeds that clinical size. And okay, this kind of sounds like, well, you can make it look like an IPMN, but the chance that it's going to be cut at that angle is quite low. But what we followed up with is when we systematically looked at all 50 blocks that we mapped, we found 22 that had these ambiguously big lesions. And then we talked to Elliot Fishman at Johns Hopkins, who looks a lot at CT and diagnoses patients in the clinic, because the second definition of IPMN is that it's 1, it's big, but 2, it can be seen in CT. And what we found was that half of these, the patients, the 22 patients with the big cysts, half of them could be seen in CT. So these actually do fit both designations of IPMN, but had been missed in the clinic because the medium used to diagnose them of H&E fails due to the fact that you don't know what angle you section tissue at. Okay. Second, I want to talk about how we combined with antibody staining to look at the complexity of inflammation at these cysts. So because of the way that we section blocks with CODA, we always hold back multiple slides. We can keep these unstained. We can also go back and add in multiplex imaging, genomics, IHC, anything else we'd like to use to map the samples. In this case, we stained leukocytes and T cells. And with this, we built what we call immunomaps. So it had been hypothesized and shown in bulk quantifications of inflammation, both in mouse models and human tissue, that Pannin is associated with inflammation. And this had kind of evolved into people saying Pannin is inflamed. And what we wanted to show with this study is that that's not the same thing. So what we found actually was if you analyze in bulk, the data would tell you that this lesion is inflamed. But actually, when we quantify it, most of the locations on this map that you see, any place that's not blue, like any of the black locations, is a cold spot, is not highly inflamed, while only the light blue is inflamed. And we designated these as cold spots and hot spots. And then we went back to the histology and showed them to a pathologist and found 2 completely distinct things going on. that the cold spots got classified by a pathologist as inactive and indolent, while the hot spots seemed histologically close to invasive disease. We followed up with this by mapping them systematically. So step one is we said, okay, imagine I analyze all these lesions in bulk. So each pannin is one data point on this plot. We found no link between a pannin's size and how inflamed it is at a local level. So this is kind of the way things would have been quantified in the past. If I take a single pannin and I ask, does its level of inflammation tell me how big it is? But when we zoom out and we instead look at the sample level, which we can only do with these big volumes of tissue and this cell-specific segmentation that we can do, we find that the patients with many pannins have a lot of inflammation, and we can fit align to that quite well. So on the left is local data looking at the individual pannin level. We see no link with inflammation, but on the patient level, we see a tight link with inflammation. Patients with a lot of pannin have a lot of immune cells. So how is this the case? What we believe is happening is the immune cells exist in the sample, but tend not to be immediately next to the lesion itself, but often distant in the acini and the collagen. Finally, and I think my slides got shuffled, but the point I wanted to make about the hotspots was we quantitatively identified the hotspots using H&E, and then followed up with an IMC panel, which gives us 50-plex immune cell labeling, and identified that these hotspot locations not only look distinct in H&E, but possess distinct immune cell types indicative of the same TME that you would find in invasive disease. So something is going on in these pannins that even though we have many, many pannins as we age, only a small subset of those cells show signs of development into PDAC. And finally, I want to show the last way that we've combined this technology with multi-omic mapping. So one of the main questions that we had as soon as we found that we develop so many of these lesions as we age is, how is it that they develop? If we have thousands of pannin, is it because one pannin initiated and then physically split into many lesions? Option two is that we independently develop all these cysts as we age. And how we could ask this question is with genomic mapping. So we used the CODA maps up on the top left to define each lesion by its anatomical connectivity. And then we combine this with LCM and DNA sequencing to look at clones in individual pannins. And just to make it complicated, in addition to mapping between lesions, we also split each pannin into 5 zones and then sequenced each of those independently to see if sometimes the same pannin could be composed of multiple clones. Okay, so to give an example, and I'll quickly explain to you what this table is showing. So up on the top, you see pannin A, B, C, and D, and those match to lesions that you can see in the video spinning on the left. On the left-hand side, you see hotspot mutations, with each of those being an independent location in the gene that could be mutated. And the 2 lines in this case show that pannin A, which is blue, had existed in 2 of the 5 zones that we sequenced in this sample and had a mutation in G12C in both of those. So what you maybe can now see is that each of these physically independent pannins possessed distinct initiating hotspot mutations, implying that they develop fully independently. So not only do we have many of these lesions as we age, but they accumulate kind of spontaneously and independently. We did find, especially in cases that had high neoplastic content, that we found many lesions with multiple combinations of mutations. And the hypothesis that we had was that this was not mixing of cells, but was 2 physically independent pannins that, because they exist in this confined ductal system, they just meet. And we did follow up with spatial-ish staining in a subset of cases, and we found that the clones do exist typically in independent ducts that just connect. And now looking a bit ahead, I'm in kind of a special spot now. I opened my lab in 2022, so a lot of what I showed today was still things that I developed when I was a student, but I'm now moving into some of my own students' continuations of what we've been looking at. So specifically in the case of pannins, we've been investigating, again, we all have so many of these lesions. What is it about a subset of pannins that indicate that they might be on the path to becoming invasive? Well, what might be a sign that this pannin is going to stay inactive until death? So we've been looking at distinct, what we call phenotypes of pannin that can be designated by both the size and shape of the nucleus and by mucin staining, and matches anatomical subtypes found in the stomach. And we've been mapping 2 levels of dysplasia in pannin to see exactly what mutations do the lesions gain and when. In the context of invasive disease, we've been looking at the initiation of metastasis. So because we do such a good job at mapping kind of physically big blocks of tissue and using those anatomical maps almost as a map to identify some unique anatomical event. In this case, we've been mapping invasive PDAC, and we want to find the moment of metastasis initiation, which we think of as almost the moment that caused the patient's death. So in this case, we've been identifying the location that the cells physically invade into the blood vessel, and then following up with Visium and IMC to look at what might be mediating this, what's allowing the cells to cut into the vessel and invade. And to do this, I think a few people have shown something analogous to this, but I thought I'd plug the method we've been developing as well. But we also have a method, like many of you do, to do automatic multi-omic alignment, which can allow us to combine things like H&E segmentation, IMC, Visium, and so on. And yeah, with that, I would like to thank you one last time, and I'm happy to take any questions.
Thank you very much for this great introduction to the pancreas and pannins.